All right, so hi everyone. I'd like to welcome you today to our science seminar from the Royal Botanic Garden Sydney. And before I start, I'd like to acknowledge the Gadigal people of the Eora Nation, the lands from the traditional custodians of the land from which we're broadcasting today, and pay our respects to their elders past, present, and emerging. So today we're really excited to hear from the work of Claire Brandenburger, who recently completed her PhD at the University of New South Wales in Angela Moll's research group. And Claire is now a digitization officer at the Royal Botanic Garden Sydney and has been with us for a year. So thanks so much, Claire, for sharing your work, and off to you.
Thanks, Hervé. Thank you so much, everyone, for hosting me, and thanks for tuning in, especially with so many new freedoms tempting you away. As Hervé said, I've been here for almost a year, and I'm working in Andre's digitization team. I'm just getting the collection imaged before I move to a shiny new home in Mount Annan. I've really enjoyed being part of things so far, and I look forward to meeting those of you I haven't met yet when we move out there and when things start opening up a bit more. The story I'll be telling you today is part of my PhD research, which I finished at the end of 2019. And it's all about the rapid evolution of an introduced plant. Now, I know a lot of you don't work on introduced plants, and you might be asking, why would one be interested in introduced plants? Well, I'm an introduced species myself. That could be part of it. I'm originally from stunning University of Cape Town on the slopes of Table Mountain. But to dissuade you from asking me any really tricky plant questions in my talk, I will admit upfront that my degrees were actually in zoology and conservation. So another good reason to study introduced species is because introduced species actually offer us an excellent recipe for rapid evolution. An introduced species is isolated from its source population, it's freed from some former selective pressures, and it's exposed to some new selective pressures. Obviously, introduced species are obviously seriously problematic for the environment. They can cost billions of dollars to manage and control. Sadly, Australia is now home to more than 3,000 introduced plant species, and many of these do affect native ecosystems, sometimes drastically, and cost money to manage. But introduced plants are also very interesting. By moving plants all over the world, we've unintentionally set up a giant experiment. And this experiment can help teach us a lot about evolution, about ecology, about genetics, and about many other things. But today we'll be looking at what introduced plants. The project which preceded my research was led by Joanna Buswell. Changes in herbarium specimens over time. And she asked the question, is rapid evolution common in introduced plant species? So she looked at almost 2,000 herbarium specimens, and she was looking for changes in morphology like plant height and leaf morphology for a number of species. And she actually found that more than 70% of her study species that had been introduced to New South Wales had changed at least one of their morphological traits since arriving in Australia. And some of these changes were quite extreme, like big changes like halving of leaf size or plant height since arrival. Now, we weren't sure if the changes that she found were evolutionary changes, because they could have been a phenotypic response to something like an environmental gradient that the plants may have been collected over time, or changes in climate over time, or changes in local land use over time. The herbarium study did try to control for as many of those things as possible, but really the only way to find out for sure would be to run common garden experiments with live plants. And that's where my research began. So in a common garden experiment, you grow plants from a native range, and you intermingle them with plants from the introduced range. And then by controlling for the environmental effects, you can then assign any differences you see to heritable evolutionary changes. And so that's what we aim to do for the South African beach daisy, Arctotheca populifolia, a beautiful study species, if I may say so myself. And it was actually one of Joanna's study specimens. So it showed an increase in plant height since its arrival in Australia in the 1930s in the herbarium specimens, and so we chose it for our study. Now, there have been previous common garden experiments where people have grown plants from across the range of the source population, side by side with plants from the introduced populations. But if you grow plants from across the whole range of the native population, you can actually introduce a large amount of unnecessary variation. And so you could end up with a result like this, where no differences can be detected between the native plants and the introduced plants. And so, you might decide that you might come across the result that introduced plants are not evolving. If you could compare the introduced plants to plants from their actual source population, however, then you would reduce a large amount of this unnecessary variation, and could help you detect if evolutionary change is actually occurring. So it's a much more precise and powerful approach, and it's the one that we aimed for in my study. So along with one of my supervisors, Bill Sherwin, in a study he was involved in, we combined genetic data, so microsatellites, with new analyses to work out where in South Africa the beach daisy was originally introduced from. And it turns out it was from a town called Arniston in the Western Cape province. And so we collected seeds from our source population in Arniston for the daisies in South Africa. And we collected seeds from 4 introduced populations up the east coast of Australia. And this spanned over 600 kilometers of the introduced range for the daisy. I grew them through 1 generation to correct for any maternal effects. And then I planted out over 400 plants. Now, before I go ahead, I just want to mention that for almost all of the traits that I measured, there was no significant difference among the values for the 4 Australian populations. And so in the majority of cases, I was able just to combine them into a single trait value for Australia. So I spent many happy hours in the UNSW glasshouse growing my plants and collecting my data over many seasons. They started out small and manageable. They grew bigger and bigger and bigger. I had to get higher and higher stakes. And, there were some days where I even feared for my life coming to uni. I didn't know what was going to be waiting for me in the glass house. But I lived to tell the tale, and I made it out of there. So, are the introduced plants evolving? Well, I divided my research into 4 areas of investigation, and I came across some surprising outcomes along the way. I first compared the morphology of the plants. And as you can see here, I uncovered some striking differences between the source plants and the introduced plants. In fact, out of the 24 morphological predictions I made based on theory and abiotic conditions, only 5 were consistent with my results. The introduced plants on the right there have evolved to be longer. They've got a sprawling horizontal body shape, and they allocate more biomass above ground. Basically, the Australians were creepier and weedier than the South Africans. I mean, obviously no surprises there, but I just had to prove it in research. And you'll also notice the very different looking leaves of the 2 groups. And you can see here a little bit what's happening. The source plants have got spoon-shaped juvenile leaves, but when they develop into adults, they get this wavy lobed leaf shape. The introduced plants retain a spoon-shaped leaf into their adult form. This is called pedomorphosis. We're not sure if this has a selective advantage or whether it's a result of genetic drift. This would have to be confirmed in more experiments. But the leaves of the introduced plants were also smaller, thicker, and less dense, all of which were opposite to our predictions. So there were a few messages to come out of this part of the research. Firstly, we showed the importance of using the source population for evolutionary comparisons. So this precise method reveals the extent of evolution that's possible in introduced plants over a short amount of time. Many trait changes were consistent with a selective advantage in the new range. Some were likely to have occurred as a result of genetic drift. The introduced plants were developing a weedier habit. And lastly, evolution in introduced plants happens, but not always in the direction predicted by theory. So next I looked at photosynthesis. Now this was the hardest area to research, but definitely my most satisfying story to tell. For this work, I based my predictions on theory and rainfall, because plants in the introduced range in Australia actually experience about 3 times as much rainfall as plants in as the source population in South Africa. And that's throughout the year. So based on plant physiological theory and rainfall, The two predictions I made were that plants in the introduced range would have a higher rate of photosynthesis and a lower water use efficiency. I collaborated with the lovely Julia Cook, and we set up two cool Lycor machines to measure a range of photosynthetic and physiological traits. That was super fun. I also looked at leaf hairs and stomata on the plants. As you can see in that picture there, the leaves of the beech daisies are covered in a very thick, furry layer of leaf hairs. And this helps protect the plants against water loss, but can also influence their photosynthetic variables. So I looked at that. And so what did I find? Were my predictions correct? The answer is not at all. In fact, I found the complete opposite. The introduced plants had actually evolved a lower rate of photosynthesis and a higher water use efficiency. And they'd also evolved an 84% greater leaf hair density underneath their leaves and lower stomatal conductance. In fact, even though rainfall is 3 times higher in the introduced range, the introduced plants had evolved changes consistent with an adaptation to drier conditions. Now, this posed quite a puzzle for us. And still, until Stephanie Creer, one of our honours students, actually went back to the beaches in South Africa and Australia to have a look. And there she discovered that the South African source plants grow on a rocky shelf, which actually traps moisture in the sand, whereas the Australian introduced plants grow on sandy beaches, from which the rainfall quickly drains. And so this highlighted 3 important points. Firstly, rainfall is not always an accurate predictor of water availability. Now, many studies do correlate climate data such as rainfall with various traits. But this work just highlighted that without detailed ecological information, we might not be able to accurately predict the direction of changes that introduced plants and other species are undergoing. And again, evolution in introduced species is happening, but not always as predicted by theory. You might be seeing a trend. So for defense traits, I measured physical defenses like ash and leaf toughness. That was fun. I got to punch holes into the leaves. I also measured chemical defenses like phenols and alkaloids in collaboration with Flo, who I'm sure is familiar to many of you. Another honours student, Martin Kim, helped me clarify the defense measurements by setting up these herbivore preference feeding trials. So I had a motley collection of 5 different herbivores, and they were presented with a choice of a South African leaf and an Australian leaf. And their preference and the amount of leaf damage they inflict were then recorded. So my predictions here were according to the enemy release hypothesis and the evolution of increased competitive ability hypothesis, or ICA. And my predictions were that introduced plants would evolve low defenses and increased herbivory. These theories predict that upon escaping their enemies in their original range, when plants arrive in a new range, they don't have to spend so many resources defending themselves. And so instead, they can put these resources into growth and reproduction, and this helps them be successful. So those were the predictions. All 6 of the defense traits that I measured showed significant differences between the 2 groups. But 3 defense traits increased in the introduced plants and 3 decreased. In fact, a meta-analysis using the defense data showed that there was no difference in the overall defense between plants from South Africa and plants from Australia. And this was corroborated with a meta-analysis using the herbivore preference trials. There was no overall preference for leaves from South Africa or Australia. So we had 3 ideas as to why this may be occurring. One possible reason we thought of was that perhaps the changes are occurring too quickly for us to measure. The beech daisy arrived in South Africa from South Africa about 90 years ago, but perhaps overall defense decreases soon after arrival and then builds up back to previous levels, in a handful of years before we have a chance to measure it. And there are some recent studies showing that changes in defense can happen as quickly as that. So that could be one reason. Another explanation for the defense changes we measured could be that different types of defense evolve in the presence of different types of herbivores. So it's theorized that introduced plants experience a shift from specialized herbivores in their native range to a more general suite of herbivores in the introduced range. and the defense changes we measured are actually consistent with this theory. In addition, there may be other selective pressures which can influence more than one plant trait at a time, including defense. So, for example, those hairy leaves that we saw in the photosynthesis, that might have evolved in a response to drier conditions, but then might also provide an increase to defense. And then perhaps another defense trait is liable to decrease. And so basically, with so many swings and roundabouts in play, it's not surprising that also in the evolution of defense in introduced species, it's occurring, but not as predicted by these overarching theories. And finally, I looked at the life history of the daisies. Now, the most striking life history change we discovered was when we tested for the ability to self-fertilize. So I got these little organza bags, and I covered the flowers to exclude pollinators and collect seeds that were produced by self-seeding. And I found that the introduced plants were spectacularly successful at making their own seeds. 104 out of 109 of my Australian plants made their own seeds, with a whopping average of about 22 seeds per plant. And in comparison, the South African plants, only 2 out of 56 made any seeds on their own whatsoever, and they just made a few each, 6 and 9. And so we have this rapid shift, just 90 years in life history, from near obligate outcrossing in the source population to predominant self-fertilization in the introduced plants. And this shift helps to explain 2 of the other main changes which we observed in the life history of the introduced plants. They've evolved a live fast, die young strategy and a weedy life history. The introduced plants have evolved a number of live fast, die young traits. Firstly, they germinate faster. So after 10 weeks, the introduced plants had germinated 99% of their viable seeds. But the source plants had only germinated 94%, and they were keeping 5% for future germination. The introduced plants also flowered a lot faster. You can see there in the orange that they actually reached their peak in flowering and started to tail off before the South African plants in purple had even begun to flower. On average, the number of days to first flower for the South Africans was 115 days, and the Australians was 85. So that's almost a month's difference in flowering time. And lastly, the introduced plants also died sooner. Only 74% of them made it to the end of the experiment, compared to 86% of the source plants. So definitely a live fast, die young strategy for the introduced plants. And the second main change was that they've developed a very weedy life history strategy. In summary, the introduced plants produced more smaller flowers, which each in turn produced more seeds, which germinated better. So they outperformed the source plants at every stage. So how is the ability to self-fertilize consistent with these 2 life history strategies? Well, if pollination is guaranteed using your own pollen, then you don't need to hedge your bets with germination time and flowering time. Instead, you would just be better off germinating and flowering as soon as possible in order to maximize your reproductive output. In addition, guaranteed pollination explains why the plants have evolved to be weedier. Because daisies have many little florets which mature sequentially, a visiting pollinator would only be able to pollinate the florets which were mature at the time. But a self-pollinating plant would be able to continually pollinate the florets and thus produce more seeds. And lastly, self-fertilizing plants can have smaller flowers too, because they don't need to be big and showy to attract pollinators. And when Steph went back to the beaches in South Africa and Australia, that's exactly what she found. There were almost no pollinators on the Australian daisies compared to the South African ones. So, are the introduced plants evolving? I hope that I've convinced you that the answer to this question is definitely yes. And so now we have something very exciting to consider. We've got a suite of morphological and physiological differences. We've got temporal separation in flowering time. We've got major changes in the reproductive strategy. And some work of an honours student showed us that we've got reduced fitness in the hybrid offspring. And so we believe that these introduced plants are now becoming reproductively isolated from their source population. In fact, with so many indications of this development of reproductive isolation, we think we might even have a new species in our hands. All the ingredients for allopatric speciation are there. The two groups are geographically isolated. They're adapting differently to local conditions. And now they're showing a number of indications for reproductive isolation. And so soon we're actually going to be applying to have Arctotheca near designated as a new Australian species. Now, I know it's controversial, and you may think that the idea of an introduced species becoming a native Australian is crazy. But some might argue that that's exactly what happened with the dingo, just over a longer time span. So our world is changing. There are now more than 13,000 plant species that have become naturalized in new parts of the world. It's too late to try and get back to some magical point in the past when there weren't as many introduced species. Some of these species are horrendously problematic. Some are pretty benign and some are even useful. But my research shows that they are evolving across multiple aspects of their makeup, and they can do so rapidly and often in ways that we can't predict. So what is the bigger picture here? Well, the bad news is that if introduced plants are evolving so rapidly, then they will only become more successful and more invasive as time goes on. The good news is that if plants can evolve so rapidly, then perhaps they will be able to adapt to new conditions created under climate change. Perhaps there's hope for plants to evolve in situ. But if you're a curious biologist, you don't really need a bigger picture. This giant experiment that we've been running by moving plants all over the world has set up some exceptional opportunities to study evolution. And I'm just thankful that I got to take one of those opportunities and help fit the pieces of a puzzle together. After all, when people ask me what I studied, I don't just say plants, I say rapid evolution. I'd like to thank Angela Moles, my primary supervisor, for her help in this research, and my other supervisors, Bill Sherwin and Stephen Bonza, all my helpers, my many helpers, my funding bodies, and you for your attention. Thank you, and any questions?
Thank you so much, Claire. This was phenomenal.
Oh,
James, would you mind stopping the recording?